I'm with the Fraser Lab at UCSF doing structural biology. I was trained mostly in crystallography, and I'm on the newer side to electron microscopy. And so today I'm going to present something that is also more on the question side, just the early stages of some work that I find really exciting. So a lot of the talks that we've seen so far have been in the size range of cells. I want to look more at this range between what chemists call small molecules and what biologists call small proteins. And this is going to be single particle imaging. So the same physical technique for a lot of the tomography talks that you've seen so far. But instead of slicing through something a whole bunch of times, we take a whole bunch of images of the same thing and then average them together. For this size scale, the single protein size scale, there are a few techniques that we have at our disposal. So nuclear magnetic resonance, NMR, is really good if we want to monitor something still in solution. We have the limitation that something can't be changing that we want to investigate on the same time scale as our experiment. So if whatever we're trying to investigate changes over the time it takes to get our data set, then we're not going to be able to answer that question. And these tend to need to be pretty highly concentrated. Not everything is amenable to that. Membrane proteins in particular, you're not going to be able to study by this method. Electron diffraction, electron microscopy and X-ray diffraction will give us structures of something that we've frozen in one conformation. So for X-ray diffraction, we let the protein or whatever particle order on its own in solution, and then it does retain a little bit of motion inside the crystal lattice, hopefully not very much. Usually we can average out one static structure from all of these instances that are oriented the same way. For electron microscopy, we let everything move around in solution and try to cryofreeze it in vitreous ice as quickly as possible so that we get all the different orientations but frozen, and then we can average together what one structure in very close to native state should look like. The disadvantage for electron microscopy is partly sample damage. The very small penetration depth means we need to create as thin a sample as we possibly can and take an image or take a data set in as few images as we can at any given position because it accumulates damage very, very quickly. And then the disadvantage for X-ray diffraction is not everything crystallizes very well. It takes a long time to optimize it. Crystallization is a black art. And so if we can't get a sample that way, electron microscopy for very many samples is going to be our only option. Some of the really cool things that we are able to do with these methods, with NMR, if you can take the same sample and collect a bunch of data sets in sequence, then you can track the kinetics of an experiment. And that's something that you can't do with these techniques that will destroy your sample all at once. With X-ray diffraction, one of the really cool things you can do is a time-resolved experiment or an actually room temperature experiment that you can't do with microscopy. You do destroy your sample, but with recent techniques like serial crystallography, you can destroy a whole bunch of crystals one after another, but get native state structures from each one. Within the limitations of what's happening inside the crystal doesn't always represent what's happening inside a cell, but you can study it at room temperature. You can start a reaction and then track particular time points. You can approximate this with electron microscopy to the degree that you can start a reaction uniformly and then trap it by cryofreezing. Anything that can be trapped that way, you can also study as some kind of approximation of a time-resolved method. So here's just a really quick video for those who are not microscopy on the protein scale experts. These are the kinds of resolutions that we're able to get now, 3 angstrom-ish. you can deposit your aqueous sample on a grid and blot it to get an extremely thin layer, hopefully still with liquid. Everything's still moving around at room temperature, and then as quickly as you can, you plunge it in liquid ethane and then move it to liquid nitrogen for imaging. During the imaging process, you get many, many copies of the same protein, so obviously you want it as pure as possible, so you get all the same thing, no contamination from other things of the same size and shape. And then you make a composite volume from all of those copies. That's why you need all the different orientations, as you heard earlier this morning. And so some of the limits we're up against, most of them are in the sample preparation area. We're looking at things like how fast you can cryofreeze and preserve most of the motion and orientations and conformations that we're interested in looking at. In terms of resolution, we're not limited by the properties of the electron beam. We're limited in terms of the detector. So for other applications, like in materials science, you can get higher resolutions under these different conditions. But for the kinds of samples we're working with, we're not able to get that sub like 2 or 3 angstrom region right now. And we are limited by the ways samples behave on the grid. Some samples like to align to the grid, and you will only get one orientation, and you can tilt the grid, but you can still only get maybe 15 degrees in each direction, which becomes a major problem when you're reconstructing the volume. So all told, most of these limitations that we're looking at right now have to do with ways we can prepare the sample, and in some cases, instrumentation. And so this last one here, flexibility, is one of the only problems that we can solve in the data processing side of things. So let's say that we want to collect a structure of a horse by single particle cryo-electron microscopy. And the question that we're trying to answer, this is a real question that people were trying to answer at one point, and these are photos by Edward Muybridge, who is trying to investigate whether during the time a horse is running, is there any point in time that the horse is completely off the ground? And you can't get this if you look at just the horse running in real time. You need to get this, these individual high-speed camera shots. And so if you were to get this averaged image, which is analogous to what we would get by standard single particle volume reconstruction approaches, it's not immediately obvious whether the horse is completely off the ground at any given point. And if we were going to analyze this the way we would analyze single particle images, we would look at the body makes this mostly stable region here. Maybe there's something going on with the head. And there look like there are a couple of leg domains, but they're disordered. They're clearly not functionally relevant. And so that's a problem. If that's the most important part that we want to study, then that's the part that we need to fundamentally treat differently in order to get that information out. And so in the last 5-ish years, there have been a number of ways that people have started to address this question of how we get motion back out of our single particle imaging data sets. And so the simplest treatment is to treat all motion as a source of noise. And if we don't want to treat all motion as a source of noise, the next simplest approach is to classify our data set into a whole bunch of different sub-data sets, get a whole bunch of different pictures of horses, and then try to analyze at the end how they relate to each other. And this has the major disadvantage, especially if you're looking at a wide range of motion. You have to cut your data set into that many individual pieces. Your signal to noise is going to suffer based on how many times you have to cut your data set. So then there's also focused classification, where you would align everything based on the horse's body, and then you would align again based on another part, and you might get this really good high-resolution image of a horse's hoof, but you still don't know how that relates to the body. And so most of the techniques that we've been able to develop until now make some assumption at some stage that there's a rigid body that moves in relation to other rigid bodies. None of these are very good approaches for looking at dynamic motions, very large motions, and none of them are really good at answering questions around if the question we want to ask is about this short-lived state. All of these techniques are good at answering questions about the major states and very poor at answering questions about transient states, which are very often the most functionally interesting. And so we're trying to answer this question with an iterative approach. And this is work that I'm doing with Alexei Rohu at Genentech, and he's co-developer of the System software. So we're building this into the System pipeline. So we start by getting our data set the standard way, getting a map and model the standard way. We refine motion into the model, and then we use that information to inform the map, and then start iterating and see how much we can improve the map itself based on that refinement of the model. So the way we go about this, we start with your experimental map and your atomic model. This is the iterative part. You can calculate what map you would get if your model is correct. You take this predicted map, and then we take the low-frequency information from the experimental map and the high-frequency information from the model. So we're trying to inform, we're trying to get a better reference volume based on the best of both worlds from those two sources of information. And then we can align micrographs to this improved volume and see how this improves over time. So this is what it looks like. This is before we have the motion part of it. This is just trying to combine those two sources of information. And the way we want to encode motion is with translation, libration, screw parameters. So this is a standard way that motion is encoded for crystallographic models. It can be a hierarchical way of describing maybe the entire molecule moves within the crystal with this one motion, and then subunits of the model move in this other motion, and you can describe either the regions or all the way down to the level of individual atoms with these parameters for translation, rotation, and screw motions. And so we want to apply those to the map itself. So these are a couple of images here. This is from when a ribosome was refined with just classification. These are two classes that came out. And you can see there's this motion that we should be able to use to connect these. And if we model this range of motion and align micrographs to the correct point on the range of motion, we hope that this blurring effect will go away and we'll actually start to get features out of that region as well. We're going to apply this to the ribosome for a positive control and a proof of concept. So our positive control is if we feed it in a model that has only one ribosome structure, and if we get out the versions of the ribosome as it's going through its ratcheting motion while it's translating RNA into protein, then we'll know that we're getting new information out that we couldn't get before. We're not just getting model bias if we start with one model and get several models that we know are biologically relevant. And then our proof of concept, the question that we really want to ask next, is we know about these specialized ribosomes that are compositionally different by individual proteins on the ribosome surface. And with or without these proteins, they have different preferential behavior. They have different sets of mRNAs that they are more likely to translate or not. And so we think this is a mechanism that has to do with flexibility. We think that depending on which proteins are present, the ribosome has different flexibility equilibria. And so if we can recover a difference between ribosomes that all have this protein present or all don't have it present, then we'll have an answer to that hypothesis. And this is we're getting ribosome samples from the Barna lab, which has done a lot of the work around specialized ribosomes, and we have microscopy with the Fraser lab at UCSF. And so I just want to thank Alexi Rohu at Genentech, Maria and Naomi at Stanford, and then Fraser Lab members. This is Jenna and John have done the microscopy with me, and this is my cat. I would love to take any questions, and if you have, I assume there are people in this room that have good ideas that I haven't even thought of, and I would love to get your input. Thank you. This one?